Hi everyone, I'm Chloe, and alongside my lab partners Tuyan and Linda, we did a fluid balance lab analyzing the effects of dopamine, angiotensin II, and high potassium. Our experiment uses Ohm's law to examine the flow of fluid through blood vessels measured by the number of drips off the frog's toes, and uses Starling's equation to examine the flow of fluid across the capillary walls in the frog, reflected by changes in the frog's weight due to water accumulating in or leaving the tissue. Moving on to methods. We began by perfusing the frog with Ringer's and dextran solution, then obtaining the controlled drip rate and weight. Afterward, we perfused the frog with the drug solution and obtained the experimental drip rate and weight for each treatment. The change in weight was calculated using the weight curve slopes.
The drugs we tested for this experiment included Ringer's and dextran with high potassium, dopamine, and angiotensin II. High potassium in the bloodstream, also referred to as hyperkalemia, is known to affect the kidneys and heart, causing symptoms such as nausea, vomiting, and heart palpitations. Increased dopamine levels in the bloodstream are typically associated with addiction and cause mental effects such as aggression and insomnia, and physiological effects such as blood vessel dilation or constriction. Increased angiotensin II levels are correlated with high blood pressure, also known as hypertension, as well as excess fluid retention. As a result, many angiotensin II receptor blockers are used as a treatment for high blood pressure. This is our patient that we will be testing the drugs described above on, and existing medical conditions include being dead, and there are no previous treatments reported.
Moving on to our results. After perfusion with the Ringer's dextran solution with dopamine, there was a decrease in drip rate by 8 drips per minute and a decrease in weight curve slope by 0.198 grams per minute compared to the control solution. The addition of dopamine showed a decrease in drip rate and a decrease in weight curve slope, therefore a negative change in slope corresponding to a decrease in weight. After perfusion with the Ringer's dextran solution with angiotensin II, there was a decrease in drip rate by 4 drips per minute and an increase in weight curve slope by 0.084 grams per minute compared to the control solution. The addition of angiotensin II showed a decrease in drip rate and an increase in weight curve slope, therefore a positive change in slope corresponding to an increase in weight. After perfusion with the Ringer's dextran solution with high potassium, there was an increase in drip rate by 6 drips per minute and an increase in weight curve slope by 0.096 grams per minute compared to the control solution. The addition of high potassium showed an increase in drip rate and an increase in weight curve slope, therefore a positive change in slope corresponding to an increase in weight. Moving on to our discussion of dopamine and angiotensin II, which stimulated the same transduction pathways. Dopamine is a naturally occurring catecholamine and an immediate precursor to norepinephrine. In the cardiovascular system, dopamine can interact with dopamine receptors, stimulate norepinephrine release, and interact with alpha and beta adrenergic receptors. Dopamine's effects depend on the perfused concentration. At low concentrations, dopamine only interacts with dopamine receptors, causing vasodilation in coronary vascular beds and others. At moderately higher concentrations, dopamine interacts with beta-1 adrenergic receptors by directly binding and indirectly by releasing norepinephrine to interact with beta-1 adrenergic receptors, ultimately causing vasodilation through the GS transduction pathway. At high concentrations, dopamine interacts with alpha-1 and alpha-2 adrenergic receptors, causing vasoconstriction and increased vascular resistance in most vascular beds through the GQ and GI transduction pathways. A decrease in drip rate and weight were observed, which corresponds to vasoconstriction through the GQ and GI transduction pathways and is explained by dopamine interacting with alpha-1 and alpha-2 adrenergic receptors on the arterioles. On the other hand, angiotensin II is a peptide hormone that binds to angiotensin type 1 receptors, or A1R, and angiotensin type 2 receptors, or A2R. Angiotensin II interaction with A1R results in vasoconstriction, while angiotensin II interaction with A2R results in vasodilation. A decrease in drip rate and increase in weight was observed, which corresponds to vasoconstriction through the GQ and GI transduction pathways and is explained by angiotensin II interacting with angiotensin type 1 receptor on the venules. Now I will get into the first transduction pathway both drugs stimulate. Both dopamine and angiotensin II stimulate the GQ transduction pathway, but through different G protein coupled receptors or GPCRs. Dopamine binds to alpha-1 adrenergic receptors located on the smooth muscle cells of arterioles, while angiotensin II binds to the angiotensin type 1 receptors on the smooth muscle cells of venules, activating the GPCRs and the GQ proteins. The activated GQ protein activates the GQ transduction pathway where the beta-gamma subunits detach from the alpha subunit, resulting in an activated alpha subunit. The activated alpha subunit then activates phospholipase C, or PLC, which then interacts with PIP2, breaking PIP2 into DAG and IP3. IP3 binds to IP3 receptors in the sarcoplasmic reticulum membrane to activate it and cause a release of calcium ions into the cytosol. DAG activates protein kinase C or PKC, which can phosphorylate L-type calcium channels or LTCCs and phosphorylate potassium channels. Phosphorylating LTCCs increases their activity and lowers their threshold membrane potential, thus increasing the ability of calcium ions to come through the channels. Phosphorylating potassium channels decreases their activities, delaying repolarization, and so LTCCs can stay open for longer. Activating IP3 receptors and phosphorylating LTCCs and potassium channels all increase the concentration of calcium ions in the cytosol. A large concentration of calcium ions binds to calmodulin, forming a calcium ion calmodulin complex. This complex then activates myosin light chain kinase, or MLCK, which phosphorylates myosin light chains. Phosphorylated myosin then binds to actin, causing cross-bridge formation. Then the myosin head bends over and a power stroke is completed, contributing to a muscle contraction. Increased calcium ions in the cytosol result in more cross-bridge formations and power strokes, and therefore there is an increased contractility and increased vasoconstriction of the arterioles for dopamine and increased vasoconstriction of venules for angiotensin II.
The second transduction pathway that both dopamine and angiotensin II stimulate is the GI transduction pathway. Dopamine binds to alpha-2 adrenergic receptors located on the smooth muscle cells of arterioles, while angiotensin II binds to the angiotensin type 1 receptors on the smooth muscle cells of venules, activating the GPCRs and GI proteins. The activated G protein activates the GI transduction pathway where the beta and gamma subunits detach from the alpha subunit, resulting in an activated alpha subunit. The alpha subunit inhibits adenylyl cyclase, which produces cyclic AMP production from ATP, resulting in decreased cyclic AMP levels. Decreased cyclic AMP results in decreased protein kinase A, or PKA activity. Typically, phosphorylation by PKA activates potassium channels, inhibits MLCK, and activates myosin light chain phosphatase, or MLCP. Decreased PKA activity results in decreased phosphorylation of potassium channels, therefore reducing their activity and delaying repolarization so LTCCs can stay open for longer. Increased time LTCCs are open results in increased calcium ions that can come through the channels into the cytosol. Calcium ions bind to calmodulin, forming a calcium ion calmodulin complex that activates MLCK. So more calcium ions available results in more MLCK activation and activity. Additionally, decreased PKA activity causes decreased phosphorylation of MLCK and MLCP, resulting in increased MLCK activity and decreased MLCP activity. Increased MLCK activity results in increased phosphorylation of myosin light chains. MLCP dephosphorylates myosin light chains, so decreased MLCP activity results in increased phosphorylation of myosin light chains. Similarly explained in the GQ pathway mechanism, phosphorylated myosin binds to actin, resulting in cross-bridge formations and power strokes. Therefore, increased phosphorylated myosin results in increased contractility and increased vasoconstriction of the arterioles for dopamine and increased vasoconstriction of the venules for angiotensin II. Now moving on to how these pathways affect the drip rate and weight for each drug. For dopamine, vasoconstriction of the arterioles decreases the radius of the arterioles. A decrease in radius while fluid viscosity and length of the blood vessel are unchanged results in an increase in resistance. An increase in resistance while pressure difference along the blood vessel is unchanged results in a decreased fluid flow through the blood vessel. corresponding to the decrease in drip rate observed after Ringer's dextran with dopamine was perfused. Vasoconstriction of the arterioles occurs upstream of the capillaries, causing a decrease of flow into the capillary, meaning decreased capillary blood pressure, or decreased PC. Only PC decreases while oncotic pressures and interstitial tissue pressures remain unchanged, resulting in a negative flux. A negative flux corresponds to a decrease in fluid flow across the capillary walls and a net water flow from tissue to blood, which results in the decreased weight observed after Ringer's dextran with dopamine was perfused. On the other hand, for angiotensin II, vasoconstriction occurred in the venules, which decreased the radius of the venules. A decrease in radius while fluid viscosity and length of the blood vessel are unchanged results in an increase in resistance. An increase in resistance while pressure difference along the blood vessel is unchanged results in a decreased fluid flow through the blood vessel, corresponding to the decrease in drip rate observed after Ringer's dextran with angiotensin II was perfused. Vasoconstriction of the venules occurs on the postcapillary side, causing a decrease of flow out of the capillary, resulting in increased PC. Only PC increases while oncotic pressures and interstitial tissue pressures remain unchanged, resulting in a positive flux. A positive flux corresponds to an increase in fluid flow across the capillary walls and a net water flow from blood to tissue, which results in the increased weight observed after Ringer's dextran with angiotensin II was perfused.
Moving on to the effects of high potassium. High potassium acts on the sodium-potassium ATPase pumps and inward rectifier potassium channels, also referred to as Kehr channels, found on the smooth muscle cells surrounding the arterioles. This results in vasodilation and an increase in capillary blood pressure, leading to increased drip rate and increased weight. When potassium is initially introduced into the frog's bloodstream, it mimics the potassium release that occurs upon depolarization of skeletal muscle. This potassium diffuses into the interstitial space surrounding the arterioles and stimulates the sodium-potassium ATPase pumps found on the smooth muscle surrounding the arterioles. This causes the pumps to begin transporting 3 sodium ions into the cell in exchange for 2 potassium ions, resulting in an overall repolarization of the cell and premature closure of the L-type calcium channels. The stimulation of Kehr channels by high potassium levels in the blood causes similar results, with the Kehr channel stimulation resulting in increased efflux of potassium from the smooth muscle cells and even more repolarization. The repolarization done by sodium-potassium ATPase pumps and Kehr channels aids in the shutting down of L-type calcium channels, reducing the amount of calcium entering the smooth muscle. The sodium concentration gradient established by the sodium-potassium pump also enhances the activity of the sodium-calcium exchanger, which pumps calcium ions out of the cell in exchange for sodium. Overall, this repolarization and hyperpolarization, along with the decrease in calcium concentrations, results in decreased calcium binding to calmodulin, which means there is decreased activation of myosin light chain kinases, resulting in decreased myosin ATPase phosphorylation. Consequently, fewer cross bridges between actin and myosin can form, ultimately leading to smooth muscle relaxation and arterial vasodilation. This vasodilation then increases the blood vessel radius, which decreases resistance and increases flow in Ohm's law, explaining the increased drip rate seen in high potassium treatment. Arterial vasodilation also increases capillary blood pressure in the Starling law, as there is now more fluid entering the capillary bed from the arterioles, causing an increase in the pressures forcing fluid out of the capillaries and into the frog's tissues. Due to this, flux in the Starling law is positive and the frog gains weight. That concludes our presentation. Thank you for listening.